Hi, this is Ufuk Kirik. With me, I have Stephen, and we'll talk to you a little bit about our work on topological imbalance in drug discovery knowledge graphs. And this is a collaboration between AstraZeneca and Mila. So what is a knowledge graph? Well, a knowledge graph is primarily a graph, and graphs are composed of nodes and edges, in this case, triples, as we call them, connecting two entities in a relationship. In the context of triples, we refer to them as the head node, in this case will be X, connected to the tail node, which in this case will be Y, through a relationship R. So HRT is also a way to refer to these. A knowledge graph is a little bit of a special case where the nodes and relationships could be of various different types. And that's the primary differentiator. Now, you want to typically try and capture whole domain knowledge, and that could come up in many different settings. In fact, you interact with a knowledge graph pretty much every single day, I would wager. Every time you search for something in Google, every time you look for recommendations, whether that's movies or books or music recommendations, or even contacts or jobs through social media, you are being directed by a knowledge graph and some inference behind the scenes. Well, in pharma, we have different tasks at hand. Most often it's target identification. And I should mention, a target is often a biological entity, gene or protein in most cases, towards which a particular drug is then developed to interact with, either to increase or decrease its function. Furthermore, you could use knowledge graphs for synthesis prediction, precision medicine, drug repositioning, or adverse event predictions. And here we show a couple of triples demonstrating different types of entities interacting in various different ways. Now, these triples can then get stitched into a much bigger graph. And the graph itself contains a diverse set of information throughout the entire domain. And the cool thing here is that they can be easily updated as information evolves. As new literature describes new types of relationships between new or existing entities, the graph can evolve as the information evolves. As such, a knowledge graph is both a database where it stores information, but it's also a graph that allows for exploration and analysis. Furthermore, graph theoretical algorithms can be applied for detecting patterns in the domain, which may not be immediately obvious. However, on the downside, they can be extremely complex to navigate. They require a particular skill set to operate as well as significant computational resources. All this domain knowledge has to come from somewhere, and graphs make use of a variety of data sources to get entity and relationship information. Now, these resources often have varied levels of certainty or confidence associated with them. Furthermore, there may be redundancies in the sense that the same biological insight, the same actual result may exist in various different platforms, various different databases in different ways, which will then lead to redundancy down the stream. Now, as you go from many different databases into one unified graph, this process is called graph composition. And this is unfortunately also where many biases in terms of topological bias, that is, may be present. In the context of target prediction, the use of knowledge graphs looks approximately like this. You start with your graph, and your graph, in loose terms, is the constituent data and how you compose them. This gets piped into a machine learning model, often a black box, which then spits out some predictions, ranked or otherwise. Now, this list of predictions may or may not be presented into human review, but then ultimately ends up in experimental validation. And in the best cases, your validation results are then put back into the graph, but we'll leave that aside for the time being. The major problem here is that experimental validation is both very time-consuming and expensive. What that means is that you need to get your review process as accurate as possible, which is also very complex and time-consuming, which means that your ranking should be as accurate as possible, which brings us to the machine learning model. And to tell us a little bit more about that, I'll invite Stephen now.
So knowledge graphs can be used as a database, as we've been discussing, and thus they can be queried like one using various types of graph-specific query language. These work much like SQL would, and you can think of the query as traversing or walking the graph structure to get to the correct answer. In this example here, we have queried the graph asking for genes that are known to associate with the disease Parkinson's. However, knowledge graphs are obviously incomplete, and you can think of a task like predicting novel targets, as well as many other drug discovery tasks, as predicting the missing edges within a graph. Machine learning is being used to try and complete these links by learning from the graph structure. These models can then be used to predict the likelihood of a certain edge. For example, looking at this green arrow on the figure on the right, what is the likelihood that the gene HP is also involved with the disease Parkinson's? From a high level, knowledge graph embedding models ultimately want to take these complex graphs and learn a meaningful low-dimensional vector-based representation for each unique entity and relationship within the graph. And this ultimately means we have two matrices at the end, one for each entity and one for each relation. So the question then becomes, how do we train a model to produce these embeddings? This is typically done via a process of graph reconstruction, where the model is trained to recreate the original training graph as effectively as possible. Essentially, the model ends up assessing the likelihood or plausibility of a given triple, with known true triples trained to be ranked above randomly sampled negative ones. And the model performance is assessed on a holdout set of some of the training triples. I'll be using a model called Transi as an example throughout the rest of this talk and throughout our results. It's one of the earliest approaches and still one of the best performing models given the correct training setup. It's also the model we focus on throughout the rest of the study. Transi exploits a simple idea that we can translate through the embedding space to get to the correct answer. So the simple idea is that we can take our head entity, for example here, the country France, we could add the relationship, capital city, and the resulting vector, once we've translated through this space, should point to approximately Paris. Or to give it a biological example, we could take the embedding for disease X, add the embedding for the relationship type associates, and it should point to approximately gene A or some gene that associates with the disease. Models use a score function, which take as input the three vectors. So the vector for the head, the vector for the relationship, and the vector for the tail. These are then combined to produce a single scalar value that represents the plausibility of the triple being true. And the score function for Transi is simply to add the relationship to the head, then subtract the tail. And then we then take the L2 norm of the resulting vector. And essentially this is measuring how close the resulting vector is to the tail vector. So a value of 0 would be the best one here, but often it's negative. What's interesting about these models is once they've been trained, they can be used to predict new edges, novel edges that don't currently exist within the graph. And this works by giving the model an incomplete triple. You can think of this as a query, and it will score the likelihood of each unique entity in the graph being the correct one to complete the triple. So here I'm querying a trained knowledge graph embedding model with the partial triple disease X associates with. The model would then score every entity, and the hope is that the top percentage of scores once sorted would be meaningful. We'll now present some of our results as well as detailing our experimental setup. So it's long been known that many real-world graphs have a heavy tail of connectivity, and we focus on degree as a measure of connectivity within this work. So many entities in our graph have more connections than others. You have these kind of hub nodes forming, very densely connected nodes, whereas most nodes have very few connections. And our biomedical knowledge graph simply reflects the underlying data that has gone through the composition stage and has been used to form it. And as we know, there are certain biases in those, right? Particular genes and certain diseases have been historically much more studied than others, thus there's more data on them, thus they end up with a high degree in the graph. So it's sort of known in the literature that there's this imbalance in many graphs. However, the effect this has on a model trained on top of these graphs has only really been hypothesized. And we set out to sort of quantifiably measure it in this study. And we make use of the heteronet dataset, which is an open source biomedical knowledge graph, and the Transi model I introduced earlier. So throughout the results, we were asking the model to propose relevant entities given a partial triple. So here we're using Parkinson's as well as various other diseases and the relationship type it associates. And as I was mentioning, due to the way knowledge graph embedding models are scored, all unique entities within the graph, including ones that don't make semantic sense based on the ontology, are slotted into place here. And what this figure is showing is that at least the model is ranking genes above other type of entities, so above drugs, above pathways, and above other diseases. So it's learning at least that genes should indeed associate with disease. Having gotten this case study out of the way, we're focused now just on the gene entities, so the top part of that previous figure. So to explain this figure, we've queried a trained model asking it to rank every gene in the graph based on how likely it thinks it is to associate with the disease Parkinson's. So each point on this graph is a gene entity, and its score or rank or plausibility, which is the value that came out of that equation we showed earlier, is plotted against the gene's degree. So how many connections the gene has within the graph based on all its relationship types. And we can see there's a very clear correlation between the two. Genes with more connections are more likely to be given a higher score by the model, which is kind of interesting, right? Especially as this trend seems to overcome the training process. So here we've colored the orange points as training genes. So these are genes that have an edge, an empirical edge in the graph to the disease. And you sort of naively expect these to be surfaced towards the top, right? So the model's being trained over numerous steps to say these ones should be ranked highly. Yet these aren't clustered towards the top, and they sort of fall roughly where you'd expect them to be based on their degree. So interestingly, this relationship holds no matter the disease we are studying. So here, and in the paper, we looked through a bunch of different diseases, and we found this relationship holds. We also wanted to just double check it wasn't down to the model. So we swapped in a bunch of different other embedding models and found the same relationship holds. We also wanted to make sure it wasn't just a target discovery problem. So we tried a bunch of different tasks, including repurposing and protein-protein interaction prediction, and we found the same relationship holds based on those other tasks. And finally, it's not just a heteronet problem, it holds across many different data sets. And I'd encourage you to check out the manuscript for much more details on all of these experiments. One additional element we wanted to investigate was if the presence of additional edge types between a given pair of entities could affect the predictive performance. So in the heteronet schema, there are 3 different possible relationship types between a given gene and disease entity. And thus far, we've been focusing on the associates edge thus far. So it could be trivial for a model to learn that if a gene disease pair is linked via an up or down regulates type, which is these are 2 primitive types of relationship between a disease and a gene, then it could be trivial to learn that it should also associate. However, in practice, this seems not to be the case. So here the green points are genes that have an up or down regulates edge type to the disease in question, so Parkinson's and melanoma. And you see that it doesn't seem to have an effect. So the green points aren't clustered towards the top of the scores here. Of particular interest is this region in the bottom left. And here there are some green points that have a single edge, and that edge happens to be to the disease of interest. And yet other single edge genes that aren't connected to the disease are ranked above it. So it seems like this doesn't really have an impact at all. I will now pass you back to Ufuk to conclude the rest of the results and wrap us up.
Thank you, Stephen. So what impact does this have in the real world? What are we talking about here? Well, those of you that have a keen eye might have seen this outlier here that's highlighted on your screen. Let me introduce you to it. This gene is polyubiquitin C, also known as UBC. It codes for 9 copies of the ubiquitin protein. Well, what does ubiquitin protein do, you might ask? The appropriate question, I believe, is what does ubiquitin protein not do? Ubiquitin is a small protein that's used to modify other proteins in the body for various different reasons. And it's definitely not within the scope of this study or within our limited time here to dive into this. However, it might be worthwhile to mention that there's various different reasons why something might be ubiquitin-related. We have highlighted 2 here that might be relevant, specifically lysosomal degradation as well as degradation via the proteasome. Now, with the current function that the current knowledge on the function of ubiquitin protein, you might think it probably has a lot of connections to genes, which it indeed does. So what we have here is the connectivity of UBC in HETUNET. And as we see, the majority, overwhelming majority indeed, of its connections is to other genes and not many to diseases. Now then the question is, does UBC make a good drug target? The models think so, because no matter which disease that we were asking it to rank, it ranks fairly high out of all the genes. But in reality, it has a single connection to a disease. And if we check what it is an upregulated edge from polycystic ovary syndrome to UBC. Now, this is interesting. We have a gene that is probably not a very good gene target, but it ranks highly. What could that be due to? We believe it might actually be down to its degree, which is particularly high within the graph. So in order to prove that, we set out to rewire its connectivity. What we take is different fractions of the edges of UBC, exclude the things that actually have, in reality, have a connection to UBC, and randomly wire it to something else. So effectively eliminating all biological knowledge. And surprisingly, no matter how much we mangle things, it still does not decrease its rank significantly. On the contrary, in some cases, it may actually significantly increase it. Now we can actually work this the other way around as well. We take the lowest ranked gene in different diseases and start randomly adding edges with various different schemas. And then we can show that we can dramatically increase its rank fairly rapidly, actually. So as Stephen mentioned, please take a look at the manuscript to dive into these more in detail. But with the remaining time, the question is, what do we do? First off, our intention is not to critique and not to criticize the methods, but rather to understand where the shortcomings are. And in that vein, it's worth noting that the standard over-undersampling methods that work on entity-level information does not cleanly map to triples. However, what we then instead advocate for is primarily awareness. So we encourage all the practitioners to investigate the topology of their graphs and also be aware of the limitations of the model of their choice. Now, further on, the predictions, maybe it's time that we move past just ranking metrics and actually indeed inspect the outputs. Perhaps sanity check them by running different alternative hypotheses like we've done here with different diseases and look out for any emerging patterns there. Last but not least, we would then also advocate for using projections of the graph with the relevant information rather than the entirety of the graph. So hopefully we have convinced you, or we have managed to show you that these graphs have skewed topological balance, and this may have substantial effect of the predictions and their utility. But that being said, we also believe that they have great promise, primarily because they provide means of organizing, integrating, and processing massive amounts of data, getting predictions very efficiently and quickly, and hopefully counter cognitive biases there within. With that said, thank you for your attention. Thanks to our colleagues and friends and collaborators, and hope to see you another day.